Our today's topic is ultrastructure and nuclear cytology during late embryo development in plants. The process of development of mature embryo from diploid zygote is called embryogenesis. In a majority of angiosperm plants, the first division of the zygote is transverse. As a result of this, two-celled proembryo is formed. The larger basal cell at the micropylar end is called suspensor cell, and the smaller one away from it termed as terminal cell or embryo cell. The embryo cell undergoes two vertical divisions and one transverse division to form quadrant and then octant stage. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. Above the cotyledonous level, embryonal axis is called epicotyle, which form shoot, and below cotyledonous level, embryonal axis is called hypocotyle, which forms radicle, as we have already discussed this part in embryo structure. Then root tip is covered with root cap, also known as calyptra. Next is ultrastructure. Plant embryogenesis can be divided into three general phases in which distinct developmental and physiological events occur. First phase is postfertilization-proembryo phase. Second is globular-heart transition phase. And the third is organ expansion and maturation phase. The first division of zygote, as I already told you, occurs in the transverse or oblique direction, resulting in the formation of two unequal cells. The basal cell is larger than the terminal cell, and terminal cell will contribute to the formation of the embryo proper, and the basal cell will form the suspensor. I can see here two-celled proembryo stage. So structure of two-celled proembryo stage. Terminal cell nucleus becomes elongated and occupies a large part of the cell. Then nucleus at this stage has electron-dense nucleolus, which contains several small vacuoles. At the micropylar end, this end, toward the micropylar end, the basal suspensor cell, extensive from the basal suspensor cell, extensive wall ingrowths appear along the cytoplasmic face of the cell wall. Short strands of rough endoplasmic reticulum and mitochondria are also observed adjacent to these ingrowths. Mitochondria are numerous. Here you can see terminal cell and the basal cell, right? This is terminal cell and here basal cell. Mitochondria are numerous, but they contain relatively very few cisternae and electron translucent area. These are mitochondria. Then, when the embryo proper reach the early globular stage of the development, the basal cell undergoes further differentiation and consists of 4 or 6 cells. I can see here early globular embryo and suspensor cell. Here, plasmodesmata occur in the common wall between the suspensor and the embryo cells, right? There are several electron-dense inclusions in the space between the plasmodesmata and cell wall. The cytoplasm of the suspensor cell. This is suspensor cell. So, cytoplasm of the suspensor cell contains mitochondria. You can see here in this diagram, these are mitochondria. Then endoplasmic reticulum. Then ribosomes. These are ribosomes. Lipid bodies. You can see here, these are lipid bodies. Small protein bodies and vacuoles. Then well-developed wall ingrowth are present along the walls of the suspensor at the micropylar end at late globular stage. You can see here these suspensor cell dishes and these are wall ingrowths, right? So cytoplasm here contains several organelles, like rough endoplasmic reticulum. You can see here rough endoplasmic reticulum. Rough endoplasmic reticulum is poorly developed and giving a swollen appearance of means various degree. Some of the vacuoles at this stage contain electron-dense deposits. Mitochondria are numerous. These are mitochondria. Mitochondria are numerous and contain few short cristae. A few dictyosomes and small protein bodies are seen in the cytoplasm. So numerous lipid bodies, these are lipid bodies, are present, and they are large and electron dense. So in this diagram, in this figure, you can see numerous small or large vacuoles. These are vacuoles. Then mitochondria, rough endoplasmic reticulum, lipid bodies. These are lipid bodies, and ribosomes. Late globular embryo stage. Here, the suspensor cell continue to enlarge. You can see here, this is endosperm, embryo, and here wall ingrowths. Because at this stage, suspensor cell continue to enlarge and most small vacuoles appeared within the cell, and a well-developed wall ingrowths were present along the walls of the embryo sac near the integumentary tapetum. In this figure, you can see these are wall ingrowths, and this is integumentary tapetum cells. So below the cotyledons are initiated, before the cotyledons are initiated, a cuticle forms at this stage over the surface of the globular embryo. But this cuticle is absent around the suspensor. In this stage, the endosperm develops, as I already told you. Here, this is endosperm, develops rapidly, and embryo cell have dense cytoplasm and abundant organelles. Then at late Hurt embryo stage, you can see here, Hurt embryo stage, this is embryo, suspensor cell, endosperm. At this stage, the suspensor cell are highly vacuolated and protruded beyond the inner integument. It continued to grow toward the micropyle, toward this side, right? But it never grew beyond the outer integument of the seed coat. During the Hurt stage of embryogenesis, the suspensor becomes large and less electron dense. Then vesicle-like structures also appear in the nuclei of the suspensor cell. The other organelles are not clearly defined at this stage. Ultrastructurally, the suspensor basal cell in some species, hair wall ingrowths, have been observed in cells presumed to have a role in absorption and translocation of metabolites from maternal tissues to the embryo proper. Mitochondria found near these ingrowths may play a role in energy-dependent transport of nutrients. The development and cytology of the suspensor in some plants, like trifolium, concluded that basal cell is an active transverse cell, absorbing nutrients from the maternal tissue, metabolizing and transporting them through the suspensor cell to the embryo proper in early stage of the embryogenesis. Then later it degenerates. I can see here no suspensor is there. So later it degenerates at the late Hurt stage of embryogenesis and is not present in the mature embryo. You can see here. So here degenerating suspensor cells showing vacuoles and electron translucent areas, and here the cotyledons embryo stage where suspensor is absent. Next is nuclear cytology. Nuclear cytology involves the study of nuclear events and structure as plant embryo develop. It includes processes like nuclear division, maybe mitosis or meiosis, changes in nuclear organization, and regulation of gene expression. Key stages includes zygote formation, early embryonic divisions, and differentiation of embryonic tissues. You can see here from this diagram, first is zygote. So zygote formation, fusion of male and female gametes of the zygote. Then nuclear divisions, mitosis and meiosis. So here you can see this divides to form two cells, terminal cell and basal cells. Then differentiation of embryonic tissues means formation of protoderm, ground meristem and procambium, then gene expression regulation, maturation and seed development, germination, etc. So first zygote means the fertilized egg that result from the joining of male and female cells. Then two-celled stays. Here embryo possesses two cells, terminal and basal cell. Then two-celled embryo and suspensor means eight-celled embryo stage. Embryo progress to the eight-celled stage. Then globular stage. Globular, where the embryo progresses to the globular stage. You can see here globular stage. This is hystorium, right? So embryo, suspensor, this is hystorium, this is globular stage. From after globular stage, then heart-shaped. Here embryo progresses to the heart-shaped stage. Then torpedo, then means embryo progresses to the torpedo stage, and ultimately embryo progresses to the mature stage. This is mature stage. Generally, diploid zygote divides into to form two-celled proembryo. First, from two-celled stage up to differentiation of organs, embryo is called proembryo. First, mitotic vision of the zygote is transverse, splitting the fertilized egg into large basal cell and then suspensor cell, right? And means close to the micropyle, and a small terminal cell or embryo cells that project into embryo sac. Then terminal cell eventually give rise most of the embryo. Basal cell continues to divide transversely, producing a thread-like structure called suspensor, which anchors the embryo to its parent tissues. Means it pushes the developing embryo into the endosperm so as to absorb nutrients from it. Terminal cell divides several times and to form spherical proembryo attached to the suspensor, and the first cell of the suspensor toward the micropylar and becomes swollen and function as hystorium. As I already told you, this is hystorium. Last cell of the suspensor at the end adjacent to the embryo is known as hypophysis, this cell. Then hypophysis later give rise to the radicle and the root cap, and cotyledons begin to form as means bumps on the proembryo stage, give the heart-shaped appearance to the embryo, then torpedo stage, and ultimately mature embryo. Thanks for watching. If you like this video, make sure to like and subscribe for more. Thank you.